going to get right in, continuing from page 27 of chapter 1, introducing Thale Cress and the bamboo shark. So I hope you enjoy. The human epigenome is designed differently from that of a plant. For when we are born, our cells are instructed via epigenetic tagging to build for different body parts, like an eyebrow or the liver. However, as mentioned above, unlike a plant, our cells lose a lot of that plasticity over time, and when a humanoid loses a finger, we are sadly unable to regrow an entire digit back. The cell memory of our stubbornly knuckle only extends to replicating skin and nerve cells, rather than the complexity of an entire appendage. The epigenome of an animal body can and will be altered within its lifetime. And the same goes for plants that must continually adapt to seasonal change of their environment. Take, for example, Thale Cress, brackets Arabidopsis thaliana, close brackets, a seasonally flowering plant that scientists have used to study epigenetics in plants for some years. Model 1. Epigenetics. The order of events for seasonally flowering Thale Cress. The internationally adopted reference for the study of plant memory through epigenetics is Arabidopsis thaliana, also known as Thale Cress or mouse ear cress. This unassuming piece of Earth's great garden is particularly attractive to scientists due to its small genome, which makes it easy to map and study. As it also exhibits a very fast life cycle, Thale Cress has made for an ideal object of exploration for over 100 years and was the first plant to have its entire genome mapped in the year 2000. My interest in Thale Cress is bound up with it being a good model for understanding epigenetics beyond the field of agriculture and botany, as I will demonstrate below. As stated, epigenetic silencing within an organism acts as a kind of memory. The study of Thale Cress has revealed that certain species require periods of extreme cold before flowering again, when conditions are suitable. Once the genome of the cress had been cloned, it became apparent that the way in which the plant was able to flower at the right time each year was through epigenetic regulation. Here is a diagrammatic example of the different chemical tags that sit above the genes, the epigenome, which regulate patterns of behaviour in Thale Cress. As with all living organisms, the genetic information of a plant, such as Thale Cress, is carried by the chromosomes in its cells. As the cell replicates, these histones and their accompanying chemical tags, brackets the epigenome, close brackets, determine the way in which the DNA is interpreted, and therefore what kind of protein, and therefore what kind of character the new cell will have. In this case, the genes which are targeted by the triggers for flowering, for example warmth and sunlight, are blocked or silenced by the epigenome. That means Thale Cress Okay, so here is a illustration of Arabidopsis thaliana, or Thale Cress. All the illustrations in the book have been kind of like rasterized out by Johan Agard, with whom I collaborated and had a really fortunate chance to work with in this project. And essentially here we have a black and white stem of the Thale Cress plant, and then at the top you have the flowering aspects in a kind of off yellow to highlight the flowering of the plant. And on the second page, so if you turn the page over on page 30, you have another one of these kind of biological diagrams. So once again, we see these kind of spirals which have been used to represent the genome. So I'll read you the caption for the spiral. The cold of winter is the epigenetic factor which turns the blocking protein off. Therefore, once each cell's genes are wound up around the histones, the blocking protein is turned off, allowing the plant to flower. So in the further unwinding of the genome, then you have like a strand of the genome, so a series of genes. And the caption says, when each cell divides and replicates, the DNA in each chromosome unwinds. And once again, you have further magnification showing the little epigenetic tags that are attached to the histones. And the caption reads, now we can see the epigenome sitting on top of the genes wound around histones. Unwound, this epigenetic tag blocks the plant from flowering. So the diagram is kind of cyclical in the sense that if you read the next caption, it would also make sense, right? The cold of winter is the epigenetic factor which turns the blocking protein off. And so you see this cause and effect relationship between the epigenome, the histone, and then the replicating apparatus of the cells as they copy and replicate the DNA, depending on which season of the year it is, producing different outcomes, one of which could be flowering. Okay, so back to the text. That means Thale Cress, like all seasonally flowering plants, will have to pass through the entire winter before flowering, after it senses the first deep drop in temperature at the onset of winter. The plant won't suddenly begin flowering if it experiences a warm day mid-winter, because the silencing occurs in every cell of the organism one by one, which takes quite some months, longer even than the event of spring. What we are witnessing in the epigenetic silencing of Thale Cress is a pattern of chemicals deployed in a repeated sequence that produces visible phenotypic change. This pattern will be passed on to the next generation of Thale Cress as a form of gene inheritance. Like plants before them, animals, brackets including humans, pass the epigenome bound up in its histones and patterns of chemical tags on to their offspring. The human epigenome has a high degree of plasticity within our lifetimes. It can also retain chemical tagging from generations before us, meaning that our behaviour today can and will affect generations of offspring further down the line. Apparently, the sins of the father will be visited upon the child of tomorrow after all. Another difference between the cell growth of plants and humans is that full mutations of the kind which express physically apparent change take enormous amounts of time in highly complex organisms such as animals. For humans, the scope for full species mutation is much more limited than that of a plant, because the way in which humans exchange genetic information is less plastic. Humans may only exchange and receive genetic materials from the gene pool through reproduction, which, compared to plants, is a highly complex process that demands an enormous amount of energy from the mother during pregnancy. Model 2. Mutation. The nomadic carpet-walking bamboo shark. Where epigenetic developmental change is a software alteration, mutation is a hard copy alteration in the DNA of an organism or virus. A number of factors can cause mutation, but it is worth noting that the cells of a human being will produce over 10,000 to 100,000 spontaneous mutations every day, every hour. Hemiciliae, I hope that's pronounced correctly, are a family of sharks that live among the northern islands of the archipelago of Indonesia, West Papua and Australia. Commonly known as bamboo or carpet sharks, the family includes several recent speciations within the early 21st century. In 2013, one of the new species, Hemicilium halamahara, was found living along the shores around Halmahera, the largest of the Maluku Islands of the Indonesian archipelago. Many years before, the naturalist and biologist Sir Alfred Wallace famously visited these islands, claiming the discovery of all sorts of natural phenomena, to which he then ascribed his own naming system, as brackets particularly white colonial settler brackets naturalists and biologists will do. Footnote 2. He considered the standard winged bird of paradise, Semiopetra wallacei, to be his greatest prize. It was allegedly only a few kilometers north of Halmahera Island that Sir Wallace came to the idea of natural selection itself via the concept of the survival of the fittest, writing a historical letter to Charles Darwin from the island of Ternate. Warmer equatorial climates have been shown to increase the chances of new life in the form of visible phenotypic mutations, where ocean currents are known to bring nutrients to the ocean's surface. Footnote 3. Plankton are the foundation of almost all life in the ocean, for in those places where currents bring nutrients to the surface, they multiply in astonishing numbers. Oceans, A Perfect Planet, BBC One, David Attenborough, 2021. Although the influence of the specific ocean currents, which bring warmer water from the Pacific Ocean into the Indian one, through Indonesia, known as the Indonesian throughflow, are not fully understood, quote, the majority of ITF Indonesian throughflow nutrient supply to the Indian Ocean is likely to support new production and significantly impact Indian Ocean biochemical cycling. Footnote 4. Ayers, J. M. and Strutton, P.G. and Coles, V.J. and Hood, R.R. and Mattia, R.J. Indonesian throughflow nutrition fluxes and their potential impact on Indian Ocean productivity, Indonesian through-flow nutrient fluxes and their potential impact on Indian Ocean productivity, Geophysical Research Letters 2014. But the reason I've selected this example of biological mutation is that the carpet shark family Hemiciliae has a habit of negating the classification systems for animal species altogether. Thus, through mutation, these carpet-walking bamboo sharks have caused embarrassing mistakes for scientists and journalists who published their discovery of apparently new species, brackets which later turn out to have been adolescents, close brackets, or claim that one particular speciation lives across a wide range of territory only for the fish to, under observation, to turn out to be a different species altogether. In a 2013 article from The Guardian newspaper, titled "Walking Shark Discovered in Indonesia," the discovery of the new species Hemicilium halmahera is celebrated by local shark scientist Fahmi from the Indonesian Institute of Sciences. We have an illustration here of the carpet walking bamboo shark. It's a sort of slank, thin fish with two fins and on its back and two sort of fins to kind of propel it along, and it's typically found with patches and blotches which are almost sort of camouflage like, and then a series of dots and sort of spots, I guess you could say, some of which start to look like smaller versions of the camouflage itself, so I can see how there's quite a lot of confusion about how to identify a species. Okay, back to the text. However, as recently as January 2020, another British newspaper, The Independent, ran the headline, "New species of shark discovered that use fins to walk along seabed," without caring to name any of the other 4 new species it's cited as having been discovered over a 12-year research project off the coast of Papua New Guinea and Australia. A complete list of the Hemicilidae shark family is not, at the time of publication, available. But one characteristic which unites them is apparent: the ability to fool their surveyors. Perhaps The Independent was reticent to name the new species precisely for this reason. The bamboo and carpet shark is so named because of A, their extended bamboo-like tails, and B, the use of their fins to propel themselves across the seabed. This characteristic has earned the Hemiciliae family the title of walking shark. As cited above, the overall species Hemicilium halmahera was first identified in 2013. Quote, "This is the 3rd walking shark species to be described from eastern Indonesia in the past 6 years, which highlights our tremendous shark and ray biodiversity," said Fahmi, a shark expert at the Indonesian Institute of Sciences. Unquote. Susanna Goldberg, walking shark discovered in Indonesia, The Guardian, 30th of August 2013. We now know that 6 of the 9 known walking shark species occur in Indonesian waters. The genes of the walking shark H. galae are small snippets of its overall DNA. As with all living organisms, background mutations occur constantly. Some of these sharks wander along the seabed and into a new subaquatic topography. Here they find a new home and become isolated from their hereditary predecessors. The cells of the organism continue to divide and replicate as usual. Only now a series of background mutations in the shark cells have generated mutations that happen to fit the new environment better than those that H. Gallet brought with them. Over time, these new genes, which, remember, are produced at random with regards to current needs, become consolidated into the new generations of the H. Gallet shark, H. halmahera. The new species, H. halmahera, has a distinctive pattern, though not distinct enough to prevent misidentification and confusion. Other species of Hemlacilli have mutated and been selected for their ability to survive in low oxygen environments. So on the opposing page, which I just read the caption to, we see two of the carpet bamboo walking sharks next to each other. These are based on the documentation which I was describing above. And one is basically blue dots and the other is with yellow, but the patches are kind of larger on the yellow one. And this is showing the mutation as it is expressed in the phenotype, in the visible aspects of the organism. With this example in mind, I want to look more deeply at a process of evolution which involves isolation, mutation, selection, and finally, speciation. It seems that Hemicillium halmahera is most closely related to Hemicillium gallet, which is native to West Papua. Due to their proximity, it is reasonable to assume that the former is a relative of the latter, H. Gallet. But I want to extrapolate the differences which do exist here as a way to discuss the role of mutation within evolution. Focusing on the visible, phenotypic differences for clarity, I observe that these two species display different patterns of spots. This difference is the result of a process of evolution that involves the expression of genes that are replicated with new variations, and this works as follows. The genes of the walking H. Gallet shark are the product of thousands of years of selection pressures. Selection pressures for survival that necessitate adaptability include mating choices by males or females, environmental factors such as predators or parasites, and, brackets, of course, one of, close brackets, the major drivers for evolutionary adaptability, the climate. The shift from H. Gallet to H. halmahera represents the type of evolutionary change spurred on by mutations that occur all the time. To recall the statistic above, each human will generate thousands and thousands of background mutations each day. Most mutations are recessive and therefore will not be expressed. Some form cancers that will go on to kill us. Aside from the selection pressures of climate, predators, and mates, another factor in the development of new species is a process of consolidation called reproductive isolation. In order for a bundle of genes within a gene pool to survive, they have to be gathered together into a species. This is what the process of speciation delivers, a scaling up of patterns of genes into whole species. Footnote 5. 1,000 Years of Nonlinear History, Manuel de Landa, 1997. The most familiar form of reproductive isolation, considered by biologists, has an external cause: geographical changes in the habitat where the reproductive communities belonging to that species live. Being is density.